So let us begin once again. Okay, I was telling you about the swinging bucket rotors. Okay, I hope with that diagram which I showed you on the whiteboard, it was clear to you. Okay, is it clear or you have any questions about it? Because I think I am now unable to draw that same diagram once again. Okay, since the meeting ended, my whiteboard has become blank. Okay, it is blank. So this particular swinging bucket rotor is a very simple device as such. Okay, the rotor looks very simple, but it has hinges, and on this hinges, the buckets can actually swing out in the outward direction. Okay, so as the speed of the rotor increases, okay, with the speed, these tubes will become more and more inclined. Okay, so from the center, they actually swing out. Okay, swing out until if the speed is very high, the tubes are almost perpendicular to the normal. Okay, they are perpendicular to the normal, and therefore the sample components are going to go exactly at the bottom of the tubes, right? And then you can imagine that if this is the placement of the tube like this, okay, perpendicular to the normal, then the sample components will have a lot of space. Okay, they will travel a lot of distance from the level of the liquid to the bottom of the tube. Is it right? And therefore the sample separation also will be maximum. The only problem with these tubes, although this cannot be considered as a major drawback of the technique, okay, but still the sample, because the samples are traveling a longer distance, maybe the time required for the sedimentation will be very much longer compared to the other types of rotors we have seen. That is the vertical and the fixed angle rotors. Is it clear to you?
Yes, sir.
Then we come, after having seen the different types of rotors, we would now go for understanding a few techniques in centrifugation. Okay, so first technique that we shall see is the differential centrifugation technique. Okay, and in this technique, we have the normal, the fixed angle type of rotor used in the centrifugation machine, and when we carry out centrifugation, the particles which are bigger in size, they will get sedimented to the bottom of the tube. Okay, and then these pellets are separated. Okay, so the larger particles which are centrifuged at a certain, say, a normal speed, you can say, not a very high speed, okay, but rather at a lower speed, the largest particles will be separated first. They are removed. Okay, the supernatant and the pellets are separated, and then the supernatant is once again added to a different tubes. Okay, and these are once again centrifuged, but this time in the second round of centrifugation, either you employ a higher speed, okay, higher RPM or higher G force, or you can carry out the centrifugation for a longer time. Okay, very often both the things are changed. That is, the time duration is also made longer, and the speed of centrifugation also is more, and therefore the particles which are little smaller than the first particles that were removed, these particles will sediment to the bottom. They are removed, and then again the centrifugation is carried out for the third time, again for a longer time and at a higher speed, so that the third type of particle will be removed, and the process is continued on and on until most of the desired particles are removed. Okay, so this is called as differential centrifugation, because we carry out different steps of centrifugation, and in every step we either change the speed of centrifugation, or we change the time, okay, or we change both the speed as well as the time. Okay, so one by one different components are separated, and an example of this is, for example, if you have a cell lysate, animal cell lysate, or maybe a muscle cell homogenate. Okay, then initially when we centrifuge it for 10 minutes at 1000 G, then the nuclei can be separated. Then you remove the nuclei and then again centrifuge the same sample at 10 minutes for a higher speed, that is
Hello, sir.
10,000
Hello.
G. Okay, so that the contractile apparatus will be removed and then
Excuse
you
me, sir.
yes.
Sir, screen नाही दिसलं sir तुमची.
Slide is not-
Okay. For me it is visible, but then is it a problem of the internet connection?
Sir, whiteboard is this 12.
Okay. I'll once again share it. Is it visible now?
Yes, sir. Yes, sir.
Okay.
Yes.
Okay. So what I was telling you was we carry out the centrifugation, and in the first round of centrifugation at 10 minutes and 1000 G, we can separate the nuclei from the muscle cells. And then again you carry out centrifugation for the second cycle. And in the second cycle at 10,000 G, okay, you are increasing the centrifugal force or you are increasing the RPM speed. And with that, you can separate the contractile apparatus. And then again, you increase the time and you also increase the speed of centrifugation for the separation of mitochondria. Okay. And then again, if you carry out the centrifugation at 1 hour and 1 lakh G, then it is possible to separate the microsomal fraction also. Okay. So what we have done is we have repeated the centrifugation and again by employing higher speed or longer time. And then we are trying to separate different components from the sample one by one. Is it clear to you?
Yes, sir.
Yes. So can you think of any advantage of this technique or any disadvantage of the technique? Before we go to that, I'll again go to another slide which shows you a diagrammatic representation of what is called as the differential centrifugation. Okay. Yeah. So can you see the mixture here? It is a cell homogenate. What is a cell homogenate or a cell lysate?
Same kind of particle.
It is a mixture of the
Sir,
cell
after-
components, intracellular components. Okay. Including maybe the cell membrane or the cell wall. If it is an animal cell, then of course there is no cell wall. Okay. So cell membrane plus all the intracellular components after lysis of the cell. Okay. So cells are lysed and all the intracellular components have now come out into the buffer or in the medium. Okay. And then this sample is centrifuged at a lower speed. Okay. So that we can separate the nuclei, we can separate the cytoskeletons, and then the supernatant is removed, the pellet is preserved, and the supernatant is once again subjected to another round of centrifugation, in which now at a higher speed, we can collect the mitochondria, we can have the lysosomes and peroxisomes. Okay, now these components are removed, and then the supernatant is once again subjected to centrifugation here, okay, in this step. And then we can probably obtain the microsomes or smaller vesicles. Now these are removed in the pellet, and then once again you subject the supernatant to the final round of centrifugation in order to obtain the ribosomes or larger macromolecules or even virus particles if they are present. Is it clear? Hello, everybody. Can you hear me?
Yes,
Yes,
sir.
sir.
Okay.
Yes, sir.
Is this technique clear
Yes,
to
sir.
you? Differential centrifugation.
Yes.
Yes, sir.
Yes, sir.
Yes, sir.
Yeah, Yogita, can you again explain this method once again?
Yes, sir.
Can you describe this method?
Yes, sir.
Yes. Please go on.
सर मला समजली पूर्ण मेथड, पण घरी थोडासा साऊंड आहे खूप.
Okay. घरातल्या आवाजामुळे तुला बोलता येत नाही का?
सर तुम्हाला ऐकायला येईल का?
हो, येतोय. आय कॅन हिअर यू. एव्हरीबडी कॅन हिअर यू.
येस सर. Sir, this method, differential centrifugation, is used in
Yes,
थोडा आवाज कमी.
नाही, we can hear you, no problem. Okay, anybody else? For the next, Aishwarya, can you explain? Okay, anyways, we'll go ahead. I think I have another slide also. Okay, same technique, but in a different way I have shown here. These are tissue cells. Okay, the tissue is obtained, it is macerated so that the cells are all separated, and then these cells are subjected to lysis. And after lysis, we have this sample mixture containing different organelles and different molecules are there. And then at a low speed for 10 minutes centrifugation, we get the pellet which is rich in nuclei as well as the cell debris. And then it is subjected, the supernatant is subjected to higher speed and longer time so that we can separate the mitochondria. If it is a plant cell, then it is chloroplasts. Okay, these are removed in the pellet, and the supernatant is once again subjected to another round of centrifugation at a higher speed of 1 lakh G and 60-minute time so that we can obtain the plasma membranes and the other internal membranes. Okay, and then the supernatant can be once again subjected to higher centrifugal force. Okay, 1,50,000 G and 3 hours centrifugation to obtain the ribosomal fraction. Is it okay? So essentially, this is a method for the differential centrifugation of different cell organelles. Is it okay?
Yes, sir.
Okay. Then here is another technique which is called as isopycnic centrifugation. Okay, or it is also called as density gradient centrifugation. So in this particular kind of a centrifugation technique, we have some material which is added to the sample, or rather the sample is added to what is called as a preformed density gradient. Okay, just as we have seen in the case of electrophoresis, we have seen gradient electrophoresis in which the gel, polyacrylamide gel, was in the form of a gradient. Okay, and we have also seen in the case of gradient elution of in chromatographic technique, what we have is a mixture or a gradient of a mobile phase, and that is used to elute the different sample components which are in the column. Okay, so similarly, we have gradient centrifugation in which we have a preformed liquid gradient, and this is made by using a chemical which is called cesium chloride. Okay, CsCl2. Okay, we can also have other types of materials such as percol, ficol, okay, or dextran, which are used to make a gradient. So what is a gradient? Gradient is different concentrations of the medium. Okay, different concentrations. So for example, we have a higher concentration of the medium towards the bottom of the tube, and we have a very low concentration at the top of the tube. So now if you put the sample in this particular tube and allow the centrifugation to take place, then what will happen? The sample components will start moving towards the bottom of the tube. But as they are moving towards the bottom, they are encountering a higher and higher concentration of the medium, okay, towards the bottom of the tube. Okay, so as the samples are moving, the samples will have a certain density. The liquid medium surrounding the particles is also having a particular density. Now, as the samples are traveling towards the bottom of the tube, they will encounter higher and higher concentration of the medium. Okay, so at a particular place, when the sample density or the particle density will exactly match the medium density, then what will happen? The sample particles will stop moving further. Okay, because as they encounter a higher density, the particles cannot overcome that. Okay, particles cannot penetrate that higher density of the medium, and therefore they will remain at a particular place in the form of a zone. Okay, we have also seen isoelectric focusing, if you remember, where we had a pH gradient in the gel. Do you remember that?
Yes, sir.
Okay, so in a similar fashion, we have a density gradient of the medium, and as the particles move in the centrifugal field, they will encounter different densities of the medium. Okay, and wherever the density of the particle and the density of the medium exactly match, the particles are not going to move further. Okay, they will remain bound to that particular area. They cannot move further. And that is how, if there are different types of particles in the sample, okay, so for example, if it is a cell homogeneate, then different organelles are there. Each organelle will have its own density. Okay, and the different organelles will form different zones in the medium. Okay, depending upon their own density. Is it okay?
Yes, sir.
Okay, so I have written here that the particles will move towards the bottom of the centrifuge tube, and when they reach their isopycnic position, that is equal density. Iso means equal, okay, same, and picnic means that refers to the term density. Okay, so wherever the density is the same, these particles will experience the liquid density that is equal to their own density, and therefore the particles will not move further. They will stop moving. Okay, and they will form a corresponding band or zone in that particular medium. Is it right? And therefore these different areas, different zones can be separated individually, and each zone will carry a particular type of particle. Is it okay?
Yes, sir.
Okay, so again, this is a diagrammatic representation. This is the medium before the centrifugation. Okay, the CSCl and the sample are uniformly distributed, but after once you carry out the centrifugation, the different particles will form different zones in the CSCl gradient. Okay, so when you start the centrifugation, the cesium chloride itself will form a gradient, and then in this gradient the different particles also will form their individual zones. Okay, each zone has stopped at a particular place, and it will not move further because the density of the medium is greater than the density of the particles, so the particles cannot move further. Okay, then there is another technique which is called as rate zonal centrifugation. Okay, so when I use the term rate, okay, just look at this term rate. It means that there is a particular rate of movement. Okay, rate of movement, and we are carrying out isopycnic centrifugation, no doubt. Okay, but in this kind of variation, what we do is we do not allow the particles to actually reach their isopycnic position. Okay, what we are doing is we are just allowing the particles to move towards their isopycnic position. Okay, so different boundaries will be formed, different boundaries of the particles. Different particles are moving towards the isopycnic position, not towards the bottom of the tube. Okay, I'm not using the term bottom because the particles are never going to reach the bottom of the tube since we have a density gradient of the medium. Is it okay? And as the particles try to reach towards their isopycnic position, we are trying to monitor the movement of the boundary by using the Schlieren optical system. Just as we have seen in the case of capillary electrophoresis, that is moving boundary electrophoresis, if you remember this. Okay.
Yes, sir.
So here we are
Yes, sir.
only monitoring the movement of the boundaries. Okay, as each boundary will move towards its own isopycnic position, the Slyron optical system is measuring the movement of the boundary, or it is calculating the rate of the movement of the different components, right? And then, as we have seen the formula earlier, we have seen the mathematical representation. Depending upon the movement of each boundary, we are in a position to calculate the molecular size of the particles, okay, molecular weight of the particles. So this particular technique is essentially used in what is called as the analytical centrifugation. This is not a preparative centrifugation, but it is analytical centrifugation. Okay, analytical ultracentrifugation, in fact. The earlier technique, that is isopycnic centrifugation, was actually a preparative centrifugation method. Okay, so each zone can be separated and then each component can be removed from that particular zone. But in this particular method of centrifugation, rate zonal centrifugation, we are only measuring the rate of movement of each component, and based on that, we can find out what is the molecular weight of the particular sample component. Okay.
Yes, sir.
Okay.
Sir,
Yes.
sir, I don't understand the difference between the red zonal and isopycnic centrifugation in terms of their principle of working.
Yes. See, principle wise, they are exactly the same. Okay. Principle wise, they are exactly the same because both are density gradient centrifugations. Okay. In both the cases, the different types of particles will move on the basis of their density. And when they encounter a particular density which is higher than their own density, the particles are actually going to stop. Okay, but in the case of rate zonal centrifugation, we do not allow the particles to reach their isopycnic position. We are just trying to monitor the movement of the boundary, the rate of movement of the boundary. Okay, and before the particles actually reach their isopycnic position, because we are determining the rate of movement is actually used to calculate the molecular weight of the different particles. Okay, if the particles, if at all the particles reach their isopycnic position, then the movement will stop. And if the movement stops, then the analytical centrifuge cannot do any analysis. Okay, because analysis is based upon the rate of the movement or speed of the movement of different boundaries. So if there is no movement, the analytical centrifuge cannot do any calculations. Is it okay?
Yes, sir. Yes, sir. Okay.
Okay. So can you see this diagram? I'll make it a little bigger for you. And here it is differential centrifugation. Okay, and here it is isopycnic centrifugation. So both the methods are compared here. In differential centrifugation, we start with a tissue sample, tissue homogenate or tissue lysate. Okay, and then at a lower speed, we can separate one component. Then we increase the speed, subject the same supernatant to centrifugation, and then we can collect another component from the sample. Okay, then we take out the supernatant, again subject it to a third round of centrifugation, and then another component can be collected, separated, and then the remaining supernatant is once again separated by using centrifugation. And in each of the step, we increase the rate, we increase the centrifugal force, or we increase the RPM, or we increase the length of time which is used for centrifugation. Okay, so different components are separated in a stepwise manner. Is it okay? And in isopycnic centrifugation, we have a gradient of the medium here, and different particles in the sample mixture will get separated in the form of zones. Okay, they will be separated in the form of zones, and these zones are removed. They can be separated. Okay, or they can be collected separately. Is it okay? How can we separate each component? Is there a question in your mind that how can we separate the individual components?
Chromatography.
Why chromatography now? The components are already separated,
Sir,
isn't it?
Pipetting out.
Component 1 is here, component 2 is here. Yes.
Sir, pipetting out.
Yes, wonderful. So using a micro pipette, okay, we can immerse the pipette to different levels, and then we can remove each component using a pipette. Or another method is we have certain centrifuge tubes, okay. The material of the tube is such that we can actually cut out the tube, okay. So we can cut out using a blade, we can cut out this portion, we can, we can cut out this portion out. We can cut this portion out. So different portions can be cut, and then each component or each portion can be analyzed for the sample. Is it okay?
Yes, sir.
Yeah, then that was about the ultracentrifuge. And I have already told you about this instrument, so I will not read this slide again. Analytical ultracentrifuge we have seen. And then I think I have a few slides only. I have about 7 to 8 minutes for this lecture to end. Before I go to these analytical methods, do you have any question?
No, sir.
Okay. So in the ultracentrifuge, there are different methods of calculating, okay, calculating the molecular weight or the sedimentation coefficient. There is one method which is called as the sedimentation velocity method. So in this particular method, the sedimentation coefficient of each sample component is determined by the analytical ultracentrifuge. And that sedimentation coefficient will be used, that information will be used for finding out the properties of the molecules. Okay, and then this is also a very dynamic method. So we can carry out certain biochemical reactions. For example, we have antigen antibody kind of a reaction, and the complex formation of the biomolecules can be ascertained, or that can be studied using this kind of a method. Okay, it can also be used for detection of conformational change. So if there is a conformational change in the molecule, then that will also lead to a difference in the sedimentation velocity. Okay, and that is how we can determine the properties of the molecule. If there is a conformational change, or if there is a binding of a ligand to a particular receptor, and things like that. Okay, so there is another method called as the sedimentation equilibrium method. So in this method, which is also similar to the sedimentation velocity method, but here, just like we have seen in the rate zonal centrifugation, we don't allow the particles to go to the other end of the tube or to the bottom of the tube. Okay, while the particles are sedimenting, okay, they will form the different zones, and different zones will there will be a kind of what is called as an equilibrium. Okay, so at the equilibrium, the particles are not allowed to pellet at the bottom, but there is a balancing between the diffusion force and the process of sedimentation. Okay, and at this particular in this particular situation, a particular concentration band of the molecules will be formed. Okay, or a boundary will be formed. And in this particular situation, again, I can tell you that there is a certain concentration profile of the sample molecules. Okay, now that particular profile is used to calculate the molecular weight of the particular sample molecules. Okay, so with this particular technique, we can calculate the buoyant density of the particles, or the density of the particles. We can find out the molecular size, or if there is a complex formation or aggregation, then we can also find out some information about the aggregation behavior of the molecules. Okay. Again, just to make the things a little more clear, I think this is a little confusing thing to understand. Okay, I'll just try to show here how the concentration profile is formed. So if you can just imagine that this is a particular boundary which is formed. This is the centrifuge tube. Okay, and this particular boundary is moving, isn't it, towards the bottom. Now, in the sedimentation equilibrium method, we are not actually allowing this boundary to go to the bottom of the tube. Okay, while it is moving, okay, when a particular stage is reached, or we can say when a particular equilibrium is reached, then there is a certain concentration profile. Okay, so what is that? Although most of the particles are concentrated here, okay, some of the particles are also lying in this particular region. Okay, so let me call it as a cone. So this particular entire region, if you analyze, okay, then the concentration here is very high, and then the concentration goes on becoming lesser and lesser. And then in this particular region, the concentration of that sample molecule is very less, isn't it? And therefore, there is a particular concentration profile which is created here. Okay, now this entire region, entire profile is used to get the information about the particle. That is the molecular weight of the particle or the sedimentation coefficient of the particles. Okay, each different component will form a different profile. Okay, and that profile is something which is very useful for getting the information about the molecules. Does it make sense to you?
Yes, sir.
I'll show here another particle. Okay, now these particles are little bigger in size. Drawing on this is a little difficult for me. Still, I'm trying it. Okay, so now if you look at this particular profile for a different molecule, okay, this profile, okay, this entire region is analyzed by the centrifuge machine or the Slyron optical system. Okay, and after analysis of this entire zone, some information about the density of the particles or the molecular weight of the particles can be found out. Is it okay?
Yes, sir.
Yes.
So that is sedimentation equilibrium. And just remember again, remember that the particles are not actually going to the bottom of the tube. They are not forming a zone at the bottom of the tube. That is sedimentation equilibrium method. Okay. So this is the optical system which is shown here diagrammatically, and that makes use of a xenon lamp. Okay, what does a xenon lamp do? Some radiation will be incident upon the sample, is it right? And wherever the particles are there, the particles will absorb light, and the amount of light which is absorbed by the particles is proportional to the concentration of the analyte. Okay, concentration of the molecules. So essentially, based upon the refractive index change in the